Good afternoon. Today I want to present your presentation about soil fungal communities as the object of astrobiological research. My name is Margarita Kriuchkova. I'm a PhD student in Lomonosov Moscow State University and junior researcher in All-Russian collection of microorganisms. Modeling of extraterrestrial environments is one of the ways to investigate the possibility of surviving of Earth-like life forms on different objects of solar system for studying the potential ways of microbial adaptation and evolution under extreme conditions. Some extreme Earth environments are similar in number of physical or chemical and chemical parameters with some of the planets and small bodies of the solar system. The native microbial communities can be used as the objects of for astrobiological research. Microorganisms and their communities in such habitats had the widest set of mechanisms for protection from effects of stress factors. For example, deserts and microbial communities from desert soils develop under the conditions of low concentration of organic matter, low humidity, extreme temperature changes, elevated isolation, and radiation. And microorganisms form universal mechanisms of cell protection in order to survive in harsh environments. According to the scientists, there was liquid water on the surface of Mars about 10 million years ago. So there was the possibility of origin or the development of life. After the loss of atmosphere, Mars became hostile for living organisms, and now the surface of Mars is exposed to different types of irradiation, extreme temperature swings, low pressure, and other factors that limited vitality preservation. Why fungi? It's widely known that microorganisms can stay alive for a long time as spores. Pure strains of bacteria and fungi survive after the exposure to high doses of ionizing radiation up to 170 kilograms. For example, Chromyces antarcticus, fungi isolated from cryptolithic community colonizing sandstone in the McMurdo dry valleys or in Antarctica. The sustainability of microorganism communities in natural substrates like soil to the impact of extreme factor is higher, but now it's not studied enough. I would like to emphasize the important role of the substrate in defense of the cells and the interactions between different species in natural communities that increase the sustainability of microorganisms. So we have harsh environment, extreme environment like desert, and fungi communities from it. As I said before, spores that can be on the surface or in subsurface of Mars, the potential spores, are affected many different exposure of temperature, pressure, and different types of radiation. And in our study, we analyze the reaction of fungi communities from desert soils to the physical impact simulated long-term presence in extraterrestrial conditions. As you can see, the everyday radiation is not so high as our experiment, because in our modeling experiment we want to use high doses of ionizing radiation that equate the presence under Mars and space conditions for 1 to 10 million years. Here you can see the methods. We use samples. The objects of our investigation were samples from 2 desert soils, gray-brown soil and gray soil from Israel. The samples were from the upper humic horizon, and the samples were irradiated. There were two main experiments, modeling experiments with gamma radiation and accelerated electrons. The samples were irradiated by high doses of gamma radiation and accelerated electrons from at low temperature and pressure, and a special climatic chamber that allowed to maintain the same level of temperature and pressure during the whole time of experiment. Here you can see this hem and some photos of this chamber and this experiment. The amount of fungal biomass in situ and its morphological structure were related by the method of direct fluorescence microscopy with chalcophrol white, ethidium bromide, and the acrogen orange dyes. Different dyes were used in order to relate not only total biomass but also live and dead biomass separately. For culturing fungi, the method of soil suspension inoculation was applied using one standard for isolation of soil microscopic fungi medium and alkaline agar with acidity similar to, not acidity, with pH similar to pH of investigated desert soils. Here you can see the scheme of experiment, not our model experiment, but the scheme of this step of experiment to see the reaction of fungi communities to this physical impact. We want to isolate as many live and culturing fungi as we can. So before inoculation, we warmed soil suspension at 52 degrees for 2 minutes to activate the germination of fungal spores, and also the petri dishes with soil suspension were cultivated at 3 different temperatures, also to isolate fungi with different temperature preferences for their growth. Now let's see the results, some of the results. First, the impact of gamma radiation on fungal biomass. Here we can see that just a second. The quantity of okay, just a second. The exposure of soil samples to 0.1 megagray gamma radiation activated fungal communities. We can see that the quantity of coloniformal units and the number of species was increased, and biodiversity also increased, as you can see here. And the most resistant fungi and dominate species was Aspergillus fumigatus. And also it's interesting to emphasize that after irradiation, we isolated some new some species that weren't observed in control samples. Now, the radiation, the exposure to 1 megagray gamma radiation led to elimination of many of species. But in this case, the most resistant species was Aspergillus niger. Also, I want to say that these two species, Aspergillus fumigatus and Aspergillus niger, are quite common, not only in desert soils, they are widespread in the world, not only in soils, but also in air. And it's not strange that they dominate after this exposure, because these two species have a set of mechanisms to from different types of stress, so they can be, and also this can be the reason for their widespreadness. Thus, exposure to gamma radiation at dose of 0.01 megagray has an activating effect on the microbiota, and exposure to a dose of 1 megagray leads to the elimination of many species. Now, let's see the results after exposure to accelerated electrons. Here we can see that the number of species was the higher in control samples, and there were a lot of dark-colored species. They are widespread in desert soil, because this dark color is because of melanin, this complex compound that help fungi to live in deserts in the conditions of high insolation and radiation. But after the radiation by isolated electrons, these doses, the number of species was almost the same, but the quantity is decreased. Also, there were some interesting points. For example, at medium in our experiment, medium doses, but still high doses of irradiation, there were many yeasts were isolated, or maybe some of the or maybe some fungi from control samples can go to this yeast form, life form. But this widespread in deserts dark-colored fungi weren't observed in after the radiation. I mean, dark-colored like this fungi, when mycelium and their quite big spores are colored. But here we also the species of genera Aspergillus were dominated after the highest doses of irradiation, and but we can see that this species here you can see the image also have dark spores that can, this melanin in their spores can help them to resist to these high doses of different types of irradiation. And if we compare these species to these species, they are dark colored, but here you can see that spores are smaller. And I think that this point also can help such fungi to survive. The effect of isolated electrons leads to the activation of spore germination of small spore species, but overall the reduction of the diversity. So to conclude, I want to say that soil fungi communities can be used as the objects of astrobiological research. And obtained results testify to the possibility of prolonged survival of eukaryotic natural communities in conditions of Mars regolith and also in the space environment inside of small bodies. Also, I want to say thank you to my supervisors and colleagues, and thank you for your kind attention.